in terms of quality oversight. And you can imagine that many of these therapeutics were built out from sort of benchtop, hands-on cell isolation and expansion technologies. And really the tools to automate and standardize those manufacturing processes are just now in development. So I think one of the things that people have been grappling with has really been consistency in manufacturing. So being able to identify lot release criteria that were rigorous enough to fail a product when needed, but were accurate and able to really advance the cell therapeutic were very important. As well, I think building in-process controls in the manufacturing process to identify the quality of cells, the health of cells, the number of cells produced, the types of products that were being secreted, the ability to monitor cell metabolism to optimize expansion platforms has all been really key. I think that there have been members of ISCT that have been quite prominent at speaking at different therapeutic societies and really advocating the need to educate the interested translational investigators as well as the emerging biotech community about quality practices in cell isolation and manufacturing, and as well as talking about regulatory issues and clinical design and issues related to translational advancement. And I know for me personally, I've had several encounters with members from USP at different meetings. I've really respected their perspective, and I've been very happy to see that they've been surveying the landscape to understand where these emerging new technologies are. And I think there's a very natural synergy between ISCT and USP. One of the other big attractions from the ISCT side is the global influence that USP has. And I think while ISCT has always had a global presence, one of the strategic initiatives is really to advance our ability to educate and guide people in developing cell therapeutics globally. I think it's a really important undertaking by ISCT, and being able to do that in partnership with a group that is an acknowledged standard center like USP is just a perfect marriage. Many of the sessions are focused on quality oversight, either around manufacturing processes, tissue acquisition and reagent sourcing, or moving all the way through clinical entry, clinical bioassays, potency assays that relate how the cells function to clinical biomarkers tied to clinically significant clinical outcome. I think it's identification of the immediate problems to solve and being able to implement an action plan to mature the industry around a specific topic, so potency assays as an example, and then to be able to feed that in collectively with USP to understand where the appropriate sort of units for standardization exist, and then to work cooperatively to define those from a scientific and technology standpoint. I think the desired output is to see a series of monographs and ongoing publications come out of USP that complement the activities and the educational processes within ISCT. One of the most important things is to find champions and people that think or identify common problems, and they don't have to be like-minded in their solutions. I think you actually want people that represent different stages of development and carry contrasting perspectives on best ways to move forward. I think out of that, we're probably going to understand and build small task forces that will identify and prioritize projects and then bring in the scientists that can best identify solutions to standardizing a certain task. So, that's going to be done with the understanding from the output from the beginning that these will result in a series of white papers to be published in Cytotherapy, or a series of monographs or standards that can then be endorsed by USP. Space is a little bit confusing currently with an inadequate description of cell types and incomplete cell characterization standards, so that either the physician or the regulatory agency or people that are interested in moving into the space from the business side can really understand the distinctiveness of a cell therapeutic approach, its distinctiveness in terms of IP and composition from competing products, and then really understand how we can set potency assessments so a physician can decide which cell type is best suited for the disease indications he's interested in working on. I think there's so much that needs to be done to characterize donor variability, potency assays that are tightly linked to the clinical space, the clinical response that we anticipate seeing. I think it's a very important element here.